Hi, my name is Yulan van Oppen, and in this presentation I'd like to talk about fast parameter estimation for ODE-based models of heterogeneous populations. Meaning we'll look into efficient parameter estimation, which involves variability. This is the main subject of my PhD project, and I've been working on it for the past 2 years with my supervisor, Andreas Milias. I'll start with a motivating example. Recently, some members of our research group used optogenetics to determine in vivo fluorescent protein maturation rates. The right panel shows a microscopy movie, and the left panel shows fluorescence data extracted from similar movies, though using a different marker. And the rates were estimated using nonlinear regression on the mean intensity trajectory, and although confidence intervals for a point estimate were given, the maturation rate variability among cells was lost. So for these kinds of data, we wanted to develop an economical way to translate variability from measurements to parameter estimates. First, we'll look into ODE-based mixed effect models, which consider multiple cells as time series with different but similar differential equations. And the gold standard to infer such models is the global two-stage or GTS method. So we'll discuss the two stages of its classical implementation first. And next, we'll get into the adaptations we have made, and finally, we conclude the presentation with some computational results to compare the accuracy and efficiency of both methods. The models of interest are nonlinear mixed effects models, and in particular those that are ODE-based. These models assume each cell has an underlying differential equation of a common form f, but with a different parameter vector beta I. The data are assumed to be noisy samples from the cell-specific ODE solutions. The parameter vectors beta I are referred to as random effects and are assumed to be distributed with mean B and covariance matrix D. In this setup, the mean B is called the vector of fixed effects. The GTS approach infers the fixed effects and the random effects covariance matrix in two steps. In the first, it estimates each cell-specific parameter vector with uncertainty Ci, and in the second stage, the spread of the first stage estimation and their individual uncertainties are considered together to recover the random effects distribution. So, let's start with parameter inference for a cell-specific time series and how it's conventionally implemented for the GTS method. Most of you will probably be familiar with this setup. We consider multiple states x with time gradients x dot and an equal number of right-hand side functions determined by parameters beta. Together they form a system of ordinary differential equations. Such systems can, for instance, be visualized with a vector field as shown here. An example is the simple model of fluorescent protein maturation, where, for instance, mRNA is generated with rate beta r and degenerates with rate beta dr. Dark protein matures at rate beta m, and so forth. The schematic on the left translates to a three-dimensional system of ODEs with 5 parameters for the rates, as shown here. Given such a model, the central question is which parameters beta fit the noisy measurement data. The first stage of the GTS method optimizes cell-specific parameters using trace matching, which is a form of nonlinear regression that minimizes the square difference between the measurements and the values predicted by our model. These predictions form the most prominent bottleneck of the optimization, because to approximate them, we generally need to rely on numerical integration approaches such as Euler, Runge-Kuzau, or Rosenbrock methods. The computational demand rapidly increases since iterative optimization schemes generally require in the order of hundreds of numerical integrations. And the procedure is also repeated for every cell individually. So next we correct for the individual estimates uncertainties in the second stage. When parameter estimates are produced using trace matching, we can approximate each error matrix Ci using the observed Fisher information matrix, or minus the Hessian of the sum of squares, which involves parameter sensitivities of the ODE solution. Next, we infer the population parameters, conditional on the true parameter values, their estimates are distributed with mean beta I and covariance matrix Ci, while the true parameters follow the unknown random effects distribution. This implies the estimates of covariance matrix D plus Ci, which leads to a likelihood that can be maximized to recover B and D. Visual explanation may be clearer. We start the second stage with a collection of cell-specific estimates beta I, shown here as a histogram with 3 cell parameters highlighted. Naively, we may infer the population parameter estimates are the sample mean and covariance matrix, with the estimated distribution shown in purple. However, when considering the individual uncertainties as well, we can correct the inflated inferred variance. The corrected approximate distribution is the GTS estimate, which is the product of an EM algorithm that maximizes the unconditional likelihood on the right. This algorithm treats the cell-specific estimates as latent variables and can be initialized from the first stage estimates in the naive distribution approximation. So let's summarize some key features of the GTS method. First of all, when properly initialized, it can produce very reliable results. However, numerical integrations are computationally demanding, and this inefficiency grows with the number of cells and parameters. Also, trace matching requires non-convex optimization, so convergence depends on initialization and is not guaranteed. So the remedy we would like to propose is to avoid solving ODE systems in the first stage using gradient matching. In biochemical systems, we can often model linearity in parameters to allow convex optimization. Altogether, this yields a much more economical approach at the price of a small accuracy loss. And next, we'll look into the adaptations we made, starting with a quick introduction to gradient matching. As the name suggests, gradients are fitted instead of direct measurements, but this requires time derivative approximations of the data. An efficient way is to use smoothing splines and their derivatives, with which we can construct an alternative sum of squares. This time we have the approximate data gradient on the left and the model gradient on the right, from plugging the smooth data into the right hand side of the ODE. Note we don't need any numerical integration for this term. But gradient matching is especially efficient in case the system is linear in parameters, so when the right hand side can be factored into a matrix function times the parameter factor. Keep in mind the system can still contain non-linear functions of the state. What's important is that linearity in parameter means the sum of squares minimization reduces to linear regression, which permits the use of convex optimization. Nevertheless, gradient matching requires all states to be measured. So to sidestep this obstacle, we use an EM algorithm where the unobserved states are the latent variables. We start from an initial parameter vector beta 0 and a known set of initial conditions x0. Next, we integrate the system to obtain the hidden states. Once we smooth the observed states, we can use gradient matching to update beta. With this updated estimate, we can integrate the system again and repeat these two steps until convergence. Although we don't avoid using numerical integration, the number of integrations still pales in comparison to non-convex optimization, especially for large models. So with the alternative first stage estimation covered, we must now efficiently compute accurate uncertainty estimates. The next part is a bit more mathematical, not essential to the rest of the presentation, so feel free to zone out for this slide. The uncertainty has to be translated from the observations y to the EM algorithm's final product beta I. The spline coefficients delta are linearly related to the data, as well as to the splines y hat they determine. However, the splines pass through the generally non-linear ODE's right hand side before appearing the expression for the estimate beta I. To add to this, the estimate non-linearly influence the approximated hidden states xi through the ODE solution, and conversely, the hidden states have a non-linear relationship to the estimates, just like the splines do. By applying several delta method approximations, we found that given the ODE solution sensitivities, we can efficiently estimate the uncertainty Ci simply as a solution to a linear system. So to summarize, we retain the overall structure of the GTS method, but we estimate the cell-specific parameters with gradient matching using linear regression instead. That means we smooth the data first before any optimization takes place. When substates are hidden, we iterate between integrating the hidden states and optimizing using gradient matching until convergence. The second stage is kept nearly identical, with the important difference in the uncertainty estimation. So the essence of our contribution consists of designing a gradient matching algorithm that can deal with hidden states, deriving an uncertainty approximation for the resulting estimate, and finally incorporating the algorithm in the GTS method for systems linear in parameters. Finally, let's review some computational results. This table summarizes the results of a small simulation study of 4 biomechanical systems. So auto-regulating protein, Michaelis-Menten mechanics, simplified protein maturation, and a bifunctional two-component system. And the second column lists whether the system was fully or partially observed. And finally, the computing times and the accuracies given by distribution distant measures of both methods are shown in the third and fourth columns. The contour plot visualizes the difference between the estimated distributions for the self-repressing protein system. So clearly we can see that major efficiency improvements can be achieved at 10 to 100-fold speedups with comparable accuracy. So in summary, we offer an economical alternative inference method for a wide range of biomechanical systems which can handle partial observations. Additionally, with adequate smoothing of the data, the obtained solutions are more predictable compared to non-convex optimization algorithms. There are 3 main challenges up ahead. First, the dependence of the solution quality on the smoothing, so improving this step is important. And second is the problem of unknown or distributed initial conditions for the hidden states. Currently, these need to be prescribed, and although they can be different for each cell, their distribution cannot be inferred. And third and final is the universal problem of identifiability. And a clear way to select an identifiable subset of parameters is not well formulated yet. And this especially holds for gradient matching. And that concludes my presentation. So thanks a lot for listening, and I'd like to give some special thanks to my supervisor, Professor Andreas Milias.